everyone, my name is Summer Rankin. I am a senior data scientist at Booz Allen Hamilton, and today I'm going to be talking about the detection of potential patient harms from clinical notes in electronic healthcare records, the Shakespeare Method. So the Shakespeare Method comes from an idea that Rosalie Bright had. Rosalie Bright is an epidemiologist at the FDA, and she wanted to use big data tools to monitor adverse events or patient harm associated with medical care. The unstructured text of electronic healthcare records tends to be really rich with a lot of information, and she also wanted to be able to detect adverse events that were attributed. So the clinician or provider actually recognized that it was an adverse event at the time, as well as unattributed, so no association. She wanted to use the natural language processing methods that were similar to the way that they investigated whether Shakespeare was the original author of his works. And so that's what inspired the name, the Shakespeare Project. And the objective here just was simply to develop a method of using EHR notes to find recognized and unrecognized potential adverse events that, and incidentally, might also uncover other anomalies in the data. So the Shakespeare Method's key innovation is that, current efforts to detect adverse events rely on a very slow process that, from the clinic, they have to first recognize that the adverse event has happened and then report it to the FDA. And by the time all of that takes place, the adverse event has already harmed, many people. And so we want to speed up that process by being able to detect things quicker, potentially even in real time. The Shakespeare Method actually unlocks clinically relevant information from text records, despite the unstructured nature of the data, the non-standard medical language, the redundant or duplicated information known as note bloat, whether or not the clinician attributes the event as an adverse event, and whether or not it's a novel medical product related harm. And so this method essentially streamlines the healthcare related adverse event detection and reporting process. So here's an example of what we might see in an electronic health record. We've got some non-standard language here in purple. Normally, there are a lot of lab values and abbreviations and any number of things that you're really not going to find in an English dictionary. And so because of this, we pre-process these notes really very minimally because for Rosalie, it was really important that we kept all of the information and we actually didn't know what was going to be relevant. So removing punctuation might actually make the lab value incorrect. And, removing stop words might actually remove an abbreviation for some kind of a medical event or a term. And so because of this non-standard language, we basically kept everything. We didn't really filter much of the text at all. Also in the yellow, a really important piece of this work was figuring out what to do with the note bloat. So typically, records may contain any kind of duplication. So a nurse or a practitioner might go in and copy a section of yesterday's notes into today's notes for one patient, and then enter their notes below that. This results in a artificial inflation or an increased importance of the earlier notes from a patient's stay in the hospital. And we didn't want that because we were using frequency-based methods. And so we developed a tool called bloatectomy, named after getting rid of the note bloat. And this is actually available on Anaconda, and you can even pip install it. And it's basically uses regular expressions to identify and highlight or remove bloated text. And so we move to let's just take an overview of what we're going to be talking about. So the Shakespeare method, the pre-processing, I'm not going to go into more detail than I already did, but you can see the paper or blotectomy for more detail on that. The first step really in this method is that you have to identify a group of interest. And this group of interest can be based on really anything. We tested it first with a treatment-based group, and then on a time period. So we tried to simulate a real-time scenario of detecting an adverse event or adverse events in general. Number two is to extract significant terms from that group of interest. And we have a couple of different ways we do that. Three is using the patients and terms from that group of interest, and then conducting unsupervised topic modeling on this particular group of patients using the vocabulary that we've pulled out of step 2. Step 4 is based on the results from step 3 to identify extreme cases. And then step 5 is to then have subject matter experts review these extreme cases. So for step 1, we're looking at creating an interest group. And we had 2 different use cases we wanted to see first whether or not it would work for a treatment-based case. And that particular treatment was blood transfusions. So we have a group of patients that did receive a blood transfusion, and that is our interest group. A group of patients that did not receive a blood transfusion, which is our comparison group. And we actually even had a group we called a gray group of patients that it wasn't clear whether or not they had received a blood transfusion, and they were actually excluded from this particular cohort. Use case number 2, we wanted to see if we could simulate a real-time analysis. And so we wanted to look at, instead of basing the 2 groups on the treatment that they received, we wanted to base it on the admission date or the time period that we were interested in. And so more specifically, we chose this particular period of time of 2007 to 2008 because it overlapped with a known heparin contamination. And there was a recall in spring of 2008 for all heparin products because of this contamination. And we wanted to see, could we detect this event happening before this, spring of 2008? So going back, we see that the periods of interest there are July 2007 to June 2008. So that's this period of interest where we think that we're going to see that heparin adverse events. And then period 2 is essentially the year prior or years prior to that. So the idea is to detect surprise events or an increasing frequency of events potentially. It also ended up pulling out administrative changes, non-medical causes of admissions, and medical practice changes in general. So let's talk about the actual steps of the technique. First, after we've decided what our 2 groups are and that we've labeled the patients accordingly, we want to then vectorize everything and then use these, basically an ensemble of filtering and classification models to pull out the most significant as well as the rare terms for the transfused group or the period of interest. And so we actually chose a naive Bayes and logistic regression model for the classification technique. And what's nice is that this technique allows you to basically get what's called feature importance out of the model. And what we can see here is the feature importance, and features here end up being terms for the non-transfused and the transfused group, which tracks with what we would expect to see the model using essentially to tell the difference between these 2 groups. And so what we do is we end up keeping the top terms or the most important terms for the group of interest. So in this case, it would be the top 5,000 terms for the transfused group. We did the same thing with naive Bayes. And then there's also a sort of a suite of filtering conditions that we also use. So one of the main things we're looking for with the filtering is just simply based on word frequency. Are there words that are specific to period 2 that are not found in period 3, and vice versa? So words that are rare within each group, as well as words that are exclusive to each group. And then we take the intersection, essentially, of all 3 of these methods to end up with, a suite of vocabulary. And that kind of depends on your computing power and what you're comfortable with. We ended up with about 40,000 features for the transfused, and somewhere around 6 or 7,000 for the time-based method. Okay, so once we've got our new vocabulary that we're going to use, we then want to take that and vectorize using only the documents of the group of interest. We then perform topic modeling, convert the topic scores to documents using the maximum topic, and pull out exceptional cases. So let's go through each of these steps. So again, first we have, for the transfused case, it was approximately 40,000 features in our vocabulary. Then we're going to take just those transfused patients, right? Just their documents. And we're going to re-vectorize everything, but instead of including, the kitchen sink like we did before, now we're only going to include these 40,000 terms. And this is important because when we did not filter anything out, we ended up with like 8 million or so different terms, because we also used multiple n-grams. And although that's possible, it's not really that feasible in terms of being able to compute easily with 8 million dimensions, right? So it helps us to reduce the dimensionality, and it's just better for computing power and time as well. We then move on to the topic modeling. So the topic modeling that we used for this study is pretty straightforward, pretty common. It's latent Dirichlet allocation. And we tried a few different ones, and this one actually gave us the best results, so that's what we chose. And what we end up with when we do topic modeling is a group of clusters or topics, if you will, that represent a group of words that tend to occur together over the corpus of documents that you're looking at. So that's why it's important that we are only looking at the group of interest for this particular step, because we're essentially trying to say what topics are going to come out of this group of, in this case, transfused patients. What are the common themes, the words that tend to co-occur? And so it's specific to the data set, which is also something that is advantageous for this particular setting. And so what latent Dirichlet allocation is going to give you is a topic and then a score for each word in each topic. And so that's kind of what you're seeing here in the chart. So for example, scores that words that tend to cluster together are all going to have a high score for the same topic, right? So aortic here belongs to probably topic 2, and not very much to topic 3 at all, and not really at all to topic 1. But dialysis actually applies to topic 1. So you're probably going to start seeing other words in that topic like kidney, renal, etc. And so then after we do, so the caveat for topic modeling is that you must choose, or for this method of topic modeling, is that you must choose a priori the number of topics or clusters that you want to include. And so this is the iterative part of this particular method. And this is where it's really important to work with your domain experts and your SMEs. I was lucky. I had 3 great epidemiologists at the FDA who I would send them, actually I'm going to show you what I would send them. I would send them basically things like this. We would also use Excel, but this is a really nice library in Python called PyLDAViz, and it allows you to visualize the results of topic modeling in a really nice interactive way. And you can even output it as HTML and just like email it to somebody. And so what you're seeing here are, the blue circles represent a topic. The size of the circle is how prevalent the topic is. And then when you hover over one of those, so in this in this static shot, number 18 that's red down there is what we're hovering over. And then what you see on the right is going to be the words that have the top scores for that particular topic. And so as we can see for this one, it looks to be some kind of a cardiac topic, right? We've got aortic and mitral at the very top there. And so this is really nice because, like I said, you have to choose that number, the number of topics that you're going to be dealing with. And it does kind of depend on the data that you're handling and what you want out of the topic modeling. What we wanted to get out of it was enough topics where we had specificity, right? We didn't want cardiac to be lumped in with like pulmonary because the topics were too few. But you also don't want to end up with so many topics that you have a ton of noise topics. You should have a few because that's the way that LDA works. It models the noise. It puts the noisy terms into its own or their own topics. But if you're ending, let's say you choose 75 topics and 50 of them are noise or junk, that's probably too many. You want to reduce it. And so that's kind of where you want to do a range. We did like 20 to 100, I think. And then we would go iteratively and the epidemiologist would come back and say, yeah, these look pretty cohesive. Let's increase it. Let's decrease the number of topics. And we landed on 45 topics for the transfused case. And I believe it was 20 topics for the time-based case because that was the less documents you have, the smaller your topic number needs to be as well. And that's what happened there. Okay, so back to our method. We then have this, I feel like people skip this step a lot with topic modeling, but once you get those topic scores for each word, you then have to apply them back to the document. And so based on those word scores and the words that are present in each document, you're going to then get a score for each topic in each document. I know it's a lot, but if you take a look at the graph here, we've got document 1 is essentially comprised of 80% topic 1 and 10% topic 2 and 10% topic 3. And so document 2 here is going to be 85% topic 2, 10% topic 3, and so forth. And so what's good to do is to assign the maximum topic back to the document. So instead of just getting every single document score, which that's useful and we do use it, you want to also take a look at the max topic for each document because that's essentially the topic for that document, right? So we would say, pretty confidently, document 1 is solidly in topic 1. Then assigning that max topic allows us to do things like look at the distribution of the maximum topic scores for each document. So this histogram is essentially showing you everybody's maximum score. Each document, or in this case, a document is a patient, each patient's max topic score. And this actually shows us something kind of interesting because what we get are people at, and so here's the overview of essentially the topic modeling process. So what we get, let's zoom in a little bit on that graph, is we have two tails essentially. So the left tail is going to show us patients that did not fit very neatly into any of the topics that we've got here. And so this essentially means that maybe they were complicated and they actually fit really well into a bunch of topics, or maybe they actually did not fit into any of these topics, which would indicate that this case is unusual, right? This patient's document is unusual. And so this is one way to kind of go through and decide, we've got 50,000 documents, which ones are we going to review? This is a great way to kind of pull those sort of unusual documents out for review. The inset here on the left side is actually one document, but it's all the topic scores for that one document. And it's a document that came from that left tail. And so as you can see, the probability there, the max is really, really small, but it's also got a tiny bit, in a bunch of different topics. So that one could be an example of just a really complicated case where they had a lot of different problems going on, or maybe they were there for a really long time, and it resulted in spreading across multiple topics. On the right side of the tail, we have people that are really solidly in one topic, and this is also a good way to kind of check whether your model makes sense, whether your topics are good, but it's also a way to investigate a particular topic as well. So we can do the same thing with an actual topic, and that's something I'm going to talk about in a second. But for the transfusion cases, the non-time-based cases, this is a great way to look at those sort of outliers and investigate further. So for the transfusion case, we ended up looking at a couple of different things. Several different pulmonary topics came out of our analysis, and we were detecting adverse events that were not attributed in the actual text. Same thing happening with cardiovascular topic. We see, so what you see on the left is our subject matter experts assign a name to that topic, but on the right are actually like the top words that are coming out of each of those topics, the top 5 words. And so that's just kind of a snippet of the results that we end up seeing. And again, looking at some people that are scoring high in these topics, some of them had a potential transfusion related adverse event. So most of the potential transfusion adverse events that were found in these notes were not attributed to be transfusion or to be related to the transfusion, meaning the clinician wasn't writing in the note, oh, this event is probably related to the blood transfusion. It's something that happens later on. So it was nice to see that our method was pulling that out. So now if we move over to the time-based case, the results are a little bit different, and how we look at the results is a little bit different as well. Because with this one, it wasn't as specific, right? So essentially what we're looking at is, you have time period 2 and time period 3. And time period 3, you can kind of treat it as like, now, or like the most recent year, if we're like pretending it's 2008. And for what we're doing, it's going to be pulling out topics that are things that are different, right, between period 2 and period 3. And so what happens when we do that is we end up looking at things that are either increasing or decreasing in period 3. And this can be any number of things. We actually ended up pulling out common things as well as uncommon things as actual topics. The thing that was related to heparin here were heart attacks. And so this hypotension right after the heparin exposure resulted in heart attacks, and we did see an increase there. We also saw an increase in things like brain trauma, brain bleeding, trauma from common reasons, as well as rare conditions. There were a few different topics that were modeling just seemed to be rare conditions. And admissions that had attributed adverse events, right? That were like this is a anaphylaxis from a drug, and it was actually written down in the text, for example. And so that was interesting and nice to see that the method was pulling that out. So some of these adverse events, interestingly, were actually taking place outside the hospital, and then was their reason for admission. And so we had both adverse events that took place in the hospital and adverse events that happened prior to them coming into the hospital, and now they've been admitted because they had some adverse reaction or event. Many attributed adverse events were not reflected in the billing codes or even the discharge notes. And this is exactly why we wanted to look at these EHR notes instead of just simply modeling on the structured data, because there's information there that, if we can access it, then we can use that to make decisions and detect things that could be at least increasing And we want to make sure that that thing that's increasing is not something that's an adverse event. So new and increased conditions were found for both common and rare conditions. Adverse events before and during hospitalization were found, and both attributed and unattributed potential adverse events were identified. The data that we used for this comes from the MIMIC data set. We used MIMIC 3, but MIMIC 4 is actually out now, and it is great. It has a ton of really rich files. They actually have recordings and sound files and all kinds of great things that go along with this data set. So if you're interested in EHR analysis, I highly recommend the MIMIC data. We also have released the entire code base for the Shakespeare method, as well as our blotectomy package, which stands on its own. So it doesn't have to be medical record. It could be any text, and you can take it through the blotectomy to detect duplicate sentences. We also have 2 publications that I've cited here. And I would just like to say a very warm thank you to my project collaborators. Rosalie Bright was our project lead. She's epidemiologist at the FDA. Kate Dowdy and Serge Block are senior data scientists with me at Booz Allen Hamilton. And Susan Bright Punt and Leanne Palmer are also epidemiologists at the FDA. Thank you very much.